of sitting next to Quaid during his sabbatical where he's at NYU. So I want to thank Quaid and Jen for this great conference and for giving me the opportunity to come and hear about your work. This is going to be a bit of a change of pace because it's going to be microbial and it's going to be pretty much, it's definitely machine learning free. But I think some of the parallels with the work, especially the cancer work, are really quite striking, and so I'm excited to tell you about this today. So in my lab, this is not working, we study adaptive evolution, and we study adaptive evolution both in the laboratory and in the natural world, using microbial systems, primarily in the budding yeast is what I'm going to talk about today. And our interest is really in connecting genotype to phenotype and ultimately to fitness. And so the work I want to tell you about today is motivated in part by this quote from Stephen Jay Gould from his book, The Wonderful Life, where he was asking this question of how reproducible is adaptive evolution. So his quote says, "I call this experiment replaying life's tape. You press the rewind button and making sure that you thoroughly erase everything that actually happened, go back to any time and place in the past, then let the tape run again and see if the repetition looks at all like the original." And Gould posited that any replay of the tape would lead evolution down a path radically different from the road actually taken, and that is that evolutionary processes are stochastic and random and contingent, historically contingent. Just anyone who's 30 or under here, a tape is something we used to listen to music on. Okay, so experimental evolution allows us to replay the tape of life many times. And in my lab, we use a device called a chemostat. This is a way of culturing microbial cells that was developed by Leo Szilard and Jacques Monod back in the middle of the 20th century. And the idea is basically that we're growing cells in this culture vessel here, and we're constantly flowing in fresh media from this media reservoir here and removing cells and culture at the same rate. And so we're constantly diluting the culture here. And this you can think of as sort of analogous to, for example, aquatic systems where there's a resident population that's constantly got some inflow and constant removal from the population. What the chemostat does, and the magic of the chemostat, is it allows the cells to grow continuously in a very defined environment. So this is the device that we use, and the importance for our purposes is that it allows us to define the selective conditions that the microbial cells are growing in with a lot of precision. And these environments are nutrient poor, and so we can control the selection by manipulating this media and having a single nutrient present at a growth limiting concentration. And so that is the selective pressure in the chemostat. And the experiments I'll tell you about today are all in the budding yeast. So what we're dealing with are asexual reproduction. We have clonal populations, so they're initially founded by an ancestral population that is genetically homogeneous. And so what we're looking at are de novo mutations that arise in the population and what their fate is in the population. We typically work with very large population sizes, which is feasible with these microbes. And so we have on the order of 1 billion individuals and a genome size on the order of 10 to the 7 base pairs. And what this means is that our mutation supply rate is extremely high under these conditions. And so we're not mutation limited at all. Everything I'm going to tell you about is in haploid cells. And the beauty of doing these evolution experiments in the lab is that we can maintain a fossil record across time. So we can be sampling the population as they're evolving in order to learn about the dynamics in terms of what's going on in the genome and also in terms of the phenotypes that are emerging in these populations. And so when we do this, here's a representative example. So here's a experimental evolution in an ammonium limited chemostat. So ammonium is a nitrogen source that yeast can assimilate and make all other amino acids and nitrogen containing compounds. And so what we're looking at is over 250 generations here. We're looking at mean population fitness relative to the ancestral strain, which is defined as fitness of 1. And what we see is that there's a rapid increase in fitness that begins to decelerate as time continues. And so here we're defining fitness as the relative growth advantage per population doubling, and we can quantify that by doing competitive co-culturing assays. And so this is something very characteristic that people see repeatedly in these long-term experimental evolution. And the question we have, of course, is what's going on at the molecular level. And to do that, we do very similar things to the sort of things you do looking at tumor evolutions and generate similar sorts of plots. So here again, we're looking across the 250 generations. And here what I've done is that I've sequenced the entire population at multiple time points here to identify the alleles that have emerged in the population and to estimate their allele frequencies. And the other advantage that we have here is that in this final population, I actually isolate clones. I can sequence those clones, and that actually allows me to identify all the mutations that have accumulated within a lineage. And so we have the piece of orthogonal information for reconstructing these lineage dynamics across time. And what you can see is very interesting is that we have new mutations that arise in the population. They begin to sweep through the population. But then in the ancestral background, we have a distinct lineage that emerges that then begins to outcompete, and mutations accumulate across time here. And so what we're not seeing is we're not seeing survival of the fittest, we're seeing survival of multiple strains that are co-resident and competing with each other. And this is a phenomenon known as clonal interference in evolution.
This is the natural mutation rate, you're not...
This is the natural mutation rate, that's right. So again, we have massive population sizes and a small genome, and so actually on average, almost every mutation is introduced into the population every generation. So
you don't get any hypermutation?
We do not under these conditions, no.
Do you ever see hypermutators in yeast?
We do, yeah. We have seen them once, but Lenski's experiments are famous for finding these hypermutators, and those experiments are actually bottlenecked, so his effective population size is much lower than the population size that we're dealing with here.
Do you check for copy number changes?
That's the second half of my talk, so yeah, it's coming. Yeah. Yeah, because I'm talking about SNPs, and I just wanted to talk about this example because it was very interesting. So here, when we look at the mutations that arise in these populations, we see 3 different mutations here that are in the exact same gene. And so this is an example of extreme recurrence. Within a single population, individually derived alleles are emerging in this single gene, GAT1. And this one was really interesting to us because GAT1 is a GATA factor. It's a transcription factor shown here that regulates nitrogen-responsive gene expression. And so this gene, GAT1, went to the nucleus, activate these nitrogen-responsive genes that are required for assimilating nitrogen sources, so getting them from the environment and metabolizing them. It's interesting little gene regulatory network because it's a GATA factor that activates these nitrogen-responsive genes, and it also activates repressors that then repress these genes as well. And so what I was showing you was a single population in which we saw 3 different mutations in the same transcription factor. And so we wanted to ask, well, how was that just a one-off in that population? And so we repeated that same selective condition in these ammonium-limited chemostats with the same founding population 3 times. 1, 2, 3 here. We see the same characteristic increases in fitness with time. And then we do whole genome, whole population sequencing, and we identify all the alleles that are at appreciable frequencies. And shown in red here is another GAT1 mutation, 2 independent ones here, and another one here. But we also had the same thought that Quaid had mentioned, that when we sequence to even 100x coverage, we're still just seeing the high frequency alleles that are present, the tip of the iceberg. And so we wanted to ask, well, what if are there other mutations there? And so we did targeted deep sequencing here to more like 1,000 to 10,000x coverage. And when we do that, targeting the GAT1 locus and some other loci, we see multiple independent GAT1 mutations that are present in the population, rising to increased frequencies consistent with selection in this environment. And so we set out to figure out how are these mutations in this transcription factor increasing fitness in this particular condition. And so here's a summary of all the mutations, the acquired mutations that we find. And this is the DNA binding domain of the GAT1, the GATA factor. And so you can see that many of them hit the same residue. We have a multitude of mutations, and the vast majority of them occur in the putative DNA binding domain of GAT1, which we also, if we go and look at natural variation, we find is under strong purifying selection out in the natural world, consistent with it being an important functional part of the protein. We can then do experiments where we ask, well, does GAT1 itself, these acquired variants, increase fitness? Because these lineages have multiple mutations, not the thousands that you see in tumors, but on the order of tens of mutations. And so to do that, we do these fitness assays where we take the lineage here that contains 5 different mutations. In this case, we measure its fitness in an ammonium sulfate limited chemostat, see that it does indeed increase fitness, and then we can isolate just the GAT1 variant and see that it also results in increased fitness and explains much of the increased fitness in that lineage. And we can do the same for this allele here and this allele here. And we see that in all cases, these GAT1 variants explain some of the increased fitness in these evolved lineages. What's important, though, is that this mutation, these adaptive mutations, are very specific to this condition, because if we go and look in other nitrogen-limited conditions, we see that, in fact, the mutations confer decreased fitness, shown in these other colors here. And so this is a case known as antagonistic pleiotropy, where they're advantageous in one condition, deleterious in another condition. And the other important result here is that if we knock out GAT1 and create a null allele, that is not beneficial. And so this is consistent with these mutations altering the function of GAT1 but not being a loss of function mutation. That's also consistent with the fact that we never saw loss of function mutations selected in our evolution experiments. We can then test, well, what are the effects of these mutations on gene expression by looking at the individual mutations and the lineages in which they emerged. And these are just the targets, known targets of GAT1. And we see something interesting, which is that about 50% of the genes are increased in expression, and surprisingly 50% of the genes are also decreased in expression. So we see this contrasting result, which we were then validated using some transcriptional reporters, where we just hooked up the promoter of MEP2 here to a GFP and found that relative to the ancestral strain shown in red here, that we see increased transcriptional output from that's attributable to these GAT1 variants. And so these GAT1 variants will increase the transcriptional output from a gene like MEP2, which turns out to be very important because MEP2 is a transporter for ammonium. So this is a logical mechanism of adapting to these environments, is to increase the amount of production of an ammonium transporter so that you can bring more of the limiting nutrient into the environment. But conversely, we also see these decreases in expression for some of the targets of GAT1, including this gene DEL80 here. And using, again, a transcriptional reporter, we show that relative to the ancestral strain, we see a great decrease in the expression from this promoter that's bound by GAT1 as well. And so we have this interesting contrast. And so we set out to try and understand why this is the case. And so GAT1 operates in a little gene regulatory network known as an incoherent feed-forward loops. The idea is that GAT1 directly activates MEP2, which is this encodes this ammonium transporter, where increased expression results in increased fitness. But GAT1 also activates DEL80, which is a repressor of MEP2. And so this is a little regulatory network that were made famous by Uri Alon's work, and that are prevalent in transcriptional networks throughout life. And so if we model what this gene regulatory network does, what it does is, GAT1, if we turn GAT1 on, it will activate MEP2, but then it also activates DEL80. And so MEP2, rather than going to infinity, its steady state level is set by this gene regulatory network, because as DEL80 turns on, it reduces expression of MEP2 here. And so if you're evolution and you want to increase MEP2 expression, what would you do? You would decrease the production of DEL80, right? Less of the repressor would result in a higher steady state expression of MEP2, and that is indeed the case. So here, if we now produce less DEL80, it represses MEP2 less. We maintain the same level of activation of MEP2, and we get an increased MEP2 steady state expression level. And so we wanted to test, is that the case? And the way that we did that is we purified these DNA binding domains from these adaptive alleles, GAT1-1 and 1-3, which I named them here. And we looked at its ability to bind the promoter of DEL80 here. And so here's the wild type with increasing amounts of the protein doing a gel shift, binding to this labeled oligonucleotide. And we see that it is able to shift the oligonucleotide with, to a good extent. But if we take these variant alleles now that we've expressed and purified, we see that there's a quantitative decrease in the ability to shift this oligonucleotide, which is consistent with decreased activation of del-80. Surprisingly, though, when we went to do the control, is that we found exactly the same was true of the MEP2 promoter. So we're trying to increase MEP2 expression. MEP2 has a much stronger promoter. It has 3 GATA sites. Here's the wild type. It's able to shift the oligonucleotide very strongly here. But these variant alleles are reduced in their ability to bind to the MEP2 promoter, consistent with reduced activation, direct activation of MEP2. But it turns out that this is actually a feature of this little gene regulatory network. That it can be the case that decreased direct activation of both the target and the repressor can result in an increased steady state level of MEP2 here. And so I've modeled that using some simple ordinary differential equations here. And here what we're looking at is the steady state expression of MEP2 scaled just to its maximal expression level. So this is what we infer is what the cells are trying to do is increase MEP2 expression in these ammonium-limited chemostats. And so this is the maximum level here. And on the x-axis, what I'm showing you is the activation, the ratio of del-80 activation to MEP2 activation. And then in blue, what I'm showing you is the strength of the direct MEP2 activation. So these are the 3 different variables here. And so if we imagine some ancestral state here in which we have maximal MEP2 direct activation, and our activation of del-80 is about 35% of that, then we have some steady state level of MEP2 that's around 30% of its maximal level. And so as I showed you, if we just decrease the extent of del-80 activation of the repressor here, we would get increased MEP2 expression. Okay? And so that's sort of the intuitive result. But the surprising result is that, in fact, there's a wide space here in which you can get decreased activation of both del-80 and MEP2, in which you still get increased steady state MEP2 activation. And so this is a surprising example in which we have sort of decreased direct activation of this target, but because of this gene regulatory network here, the net result is increased expression under these conditions. So to go back to these results, what we were looking at here is these GAT1 variants that were rising to appreciable frequencies in the population. But the close observer would see that ultimately there is another allele shown right here in blue that sweeps to high abundance in these populations. And that's one of these different class of alleles. This is a copy number variant. So this is MEP2, again, increasing expression of MEP2, but in this case by expanding the copy number size of the MEP2 gene itself. And it actually ultimately outcompetes these GAT1 variants. And this wasn't a big surprise to us because work from our lab and from a number of labs has shown that copy number variation is a recurrent result of selection in these nutrient-limited chemostats that happens with great specificity. So here I'm showing you 4 different genomes. This is using the ancient technology of array comparative genomic hybridization. These are the 16 chromosomes of yeast. This is a single lineage that was evolved in a proline-limited environment. And we see this clear signal of an amplification right here that includes the PUT4 gene, which encodes a proline transporter, and so which makes a lot of sense here. We see the same result here in urea limitation. We get a local amplification. It includes the urea transporter. Here's another limitation, glutamine limitation. This amplification here includes the GAP1 transporter. And here's another case, and we have other events that are happening in the background as well. But this is something we've seen again and again in these long-term evolution experiments in these nutrient-limited environments when we look at the end. But the problem for us has always been that we have no idea what's going on before that, and that's because for us it's been a challenge to be able to identify these copy number variants until they are present at high frequency in these populations using sort of short read sequencing or array CGH type approaches before. And so we wanted to develop a method that would allow us to be able to track these copy number variants as they emerge in the population and allow us to characterize the dynamics with which they're moving through the population in the same way that we can do with the SNP allele frequencies using deep sequencing. And so we had the very simple idea is, what if we could use a phenotypic reporter for a copy number variant? And the idea simply is, if we duplicate a gene that encodes a fluorescent protein, do we see a quantitative increase in the signal? And so we tested that just by engineering strains that had either 1 or 2 copies of a fluorescent M-citrine gene, which is just like GFP. And you can see that there's a clearly distinct signal when we run these cells through a flow cytometer. Similarly, in a diploid cell now, we engineered 1, 2, 3, and 4 copies. And again, we can distinguish the number of copies of a GFP gene on the basis of the fluorescent signal. And so the simple idea is, well, now that we know that when we do these evolution experiments, we frequently see these copy number variants at these transporter gene loci, let's just stick a GFP right at that locus. And given that typically when we see copy number variants, they include a much larger region than just the gene itself, what we expect then is if we have that gene sitting next to a potential target of copy number variation, that when that locus duplicates, we'll get a duplication and we'll be able to distinguish that signal. And so we tested that here looking by creating this construct. And so this construct in this case is this gene GAP1, which is a general amino acid permease, which transports glutamine. And so here what I'm showing you is if we stick that fluorescent protein somewhere in the genome that the cell doesn't care about, if we get a copy number variant, then over 200 plus generations we see that it's very stable. If we stick 2 copies of that gene in 2 different places in the cell that we don't expect to undergo copy number variation, it's very stable. But if we stick that GFP gene right next to the GAP1 gene, that in a glutamine limited environment we expect with some frequency to be amplified and undergo selection, we see a clear signature here where that single copy of that gene has duplicated and has swept through the population. So this allows us to do exactly what we wanted, which is to be able to follow the dynamics now in real time of these copy number variants just by running cells through the flow cytometer and counting fluorescence. And so we undertook an experiment now here where we took that reporter, that identical reporter next to GAP1. We're looking over 250 generations. We're looking at 30 different populations here. And 10 of those populations are here in glutamine limitation. And we can see very clearly that they start out with a single copy. And at some point, there's an amplification that occurs and they increase in their copy number variation at the GAP1 locus in all these populations. At the glucose, when they're growing in a glucose poor environment, we don't expect an amplification at this locus to be advantageous. And indeed, we see that it's relatively stable. And in a different nitrogen environment, we expect, and based on previous studies, we've found sometimes losing that GAP1 gene is beneficial. And in fact, we see evidence that there's loss at the GAP1 locus. And in fact, here's a clear example where the entire locus has been deleted and that has swept through the population. And so here we're just looking at the fluorescence, but we can use gating to be able to identify single versus duplicated copies. And we see remarkably reproducible results where we now quantifying on the y-axis here the proportion of cells that have a copy number variant at the GAP1 locus. This is in these glutamine-limited environment. And we see that by about 75 generations in all these populations, we see amplification, de novo amplification alleles that have been introduced into the population that sweep through the population with very rapid kinetics. It turns out to be extremely reproducible initially, and then later things get more interesting. And so, being able to use this now tells us, okay, so these mutations are clearly, they have a very strong selective advantage. They're occurring at some high rate, and so they're actually behaving in a kind of deterministic way in the population, emerging and being selected. The trivial explanation for this is that in this founding population, there had been some common allele that was already present that's just undergone selection with the same kinetics in all these populations. But if we do whole genome sequencing, and here I'm just showing you 2 different individual lineages that we pulled out from these two different populations. This is the GAP1 locus. We just use read depth to be able to identify copy number variants. And we can see clearly here that there are two distinct alleles on chromosome 11 where GAP1 lies that differ between these two populations, suggesting that this trivial explanation is not the case, that in these populations we're getting individual de novo amplifications at this locus that we're able to detect. And in fact, we've detected many different clones that have different levels of copy number variant. And it turns out that the total fluorescence for each of those clones is in good agreement. But so this tells us that between the populations, these are independent amplification alleles that have emerged and swept through the populations with very similar dynamics. But what about within a population? So we would like to know, is there just a single allele that emerges and sweeps with predictable dynamics? Or are there actually multiple alleles, multiple independent copy number variants that have emerged within a population and that are sweeping through the population? And that turns out to be a hard problem because a lot of these copy number variants are actually, if you look at independent samples, they're actually recurrent. And that's because there are repetitive regions of the genome that can promote the generation of these copy number variants. And so it's actually not easy to be able to look at the genome sequences and be able to say, well, these are independently derived alleles. Because even if you see the identical allele, they could be just a recurrent result, a recurrent mutation. And so the approach that we took is that we generated a library of random molecular barcodes. So into our strain that has this copy number variant reporter, we threw about 100,000 random DNA sequences at a specific locus. So these are molecular barcodes. And then we replayed these evolution experiments, again looking at the fluorescence, which we're looking at on the y-axis over here. And we can see that, again, we're getting a signal of copy number variants emerging in the population and sweeping through the population. And then at each of these time points, these 3 time points here, we sorted out this subpopulation that had these copy number variants using a fax machine. And we sequenced these molecular barcodes. And so the expectation would be if it was a single allele, we'd see a single molecular barcode. But in fact, what we see here are at least 8 independent molecular barcodes, suggesting that there are 8 independent copy number variant alleles in this single population that we're then able to track over time. And we can see that, in most cases they're expanding in their frequency, although in some cases they're decreasing. So there's competition between these independent copy number variant alleles. In gray here are molecular barcodes that we detect at low frequencies that are either copy number variants that were ultimately lost from the population, or they're false positives that came through in our sorting. And so,
This includes questions, by the way.
yeah. So I'm going to conclude there that we learned about evolutionary dynamics. I told you something about the molecular basis of adaptation, that we see cases of both transcription factors mediating adaptive evolution in surprising ways. And we also see that increasing copy number variation is a recurrent means of adaptation in these environments. And so I'll conclude there by thanking the funding and the members of my lab, and thank you again for listening. And I'm done. Yeah?
Does the MEP2 also have negative pleiotropy, or is the transporter duplication not as negatively affecting fitness in other environments?
So previously we have found that these amplification alleles do also show antagonistic pleiotropy. So if you put them in some other environment in which they're either neutral or have a fit, they can confer a fitness cost, and so they can be lost under those conditions. Yeah, I think this tool allows us now to be able to go and address that question much more rigorously.
Could you just put your conclusion slide back while you're answering questions?
Yeah, sorry. Yeah, that's more interesting than our hike. Mike.
Considered Phil Heater's sin, as he studies preconditions for chromosomal instability and using your system, would the dynamics stay the same, you think?
No, I would imagine they'd be totally different. I mean, this is just, the problem here is that we don't know what the background mutation rate is of these copy number variants. The estimates differ by 5 orders of magnitude in yeast for trying to determine that. And they're probably highly variable, the same sort of variation that we see in SNP mutation rate across the genome. And so I have no idea what the rate at which these GAP1 variants are produced in the absence of selection. They were under strong selection here. But it seems clear to me that the supply rate is really high. These events are occurring presumably at some high rate that we'd like to know.
Do you ever see much aneuploidy in the types of conditions that you're looking at?
Yes. Yeah, and that's another class. So the copy number variants, we can now begin to, now that we can pull them out, isolate them by using this reporter, we can then quantify, the fraction that are local duplications and the fraction that are aneuploidies, because aneuploidies have exactly the same signal. A whole genome duplication would not, though, because you just, we normalize to the signal would be the same in that case. But in these evolution experiments, we frequently see aneuploidies, we frequently see whole genome duplications, but we've never seen chromothripsis or chromoplexy, but maybe we just weren't looking right. That's why I was asking about how you actually find them. I'm worried they're all in my unmapped reads, but I need to go and check.
But have you done like have you run structural variant reconstruction algorithms on your data?
Yeah, so now I feel like, we have the ground truth. We have a reporter that tells us we have a copy number variant of that locus. We sequence it, we look at the read depth, it's clear, and we've run all 200 algorithms, and they're all crap, is my feeling. And so that's why I'm very interested to talk to you about how to do that, because most of them don't get these amplifications that we know are there. So, but now we can begin to try and learn why that is the case. Yeah, or I can learn from you. Yeah.
In your gel shift assays, do you ever see one DNA of MEC2 promoter being bound by multiple transcription factors? I mean, what I'm getting to is potential role of cooperative action of multiple transcription factors on it.
Right. So I mean, the gel shifts are all in vitro. So. And we just have the GAT1 protein there, but there's certainly likely to be cooperative and competition between these.
Binding to one promoter, and they kind of having a different output, a different expression output than the sum of two individual GAT1s binding to.
All right. Yeah. No, we haven't seen that. Yeah. Interesting.
So, I mean, I find it very impressive how